pleasure to be here to tell you about some of our work. I'd like to thank Stephen for this invitation. Stephen, wherever you are, is somebody that I met many years ago. Actually, is where we started this work while I was still in Chris Dobson's lab, the late Chris Dobson in Cambridge. And so it was nice to be able to take it down to UCL. And I think kind of both myself and Fleming, and I'm quite proud of the NMR facility that we've set up actually at UCL, because I think it's quite a vibrant facility, and really demonstrates the power of the technique. So I'm going to be telling you about the work that on the ribosome that we've been looking at, and especially, I guess, given that as we all know, all proteins are synthesized on ribosomes. This 2.5 megadalton macromolecular machine needs no introduction to the LMB for many reasons, but essentially, as the nascent polypeptide synthesized here at the peptidyl-transferase center emerges from this 100 angstrom long exit tunnel, it has its first opportunity to sample conformational space and actually can begin to fold while the chain is still attached to the ribosome. There's a search for conformational space here. And this is the process of co-translational protein folding. And so while kind of, as we know, the ribosome, and in particular mechanisms at the peptidyl-transferase center are very well understood structurally and biophysically. Actually, the nascent polypeptide is much less well understood. The range of cryo-electron microscopy, and prior to that, the X-ray crystallographic structures were able to reveal the strong density here, while the kind of the nascent polypeptide on its ribosome, the product of the ribosome, is actually poorly seen through its highly dynamic nature. Now, there've been many over recent years, this area has redeveloped a resurgence in trying to understand the process of folding as it occurs on the ribosome. Kind of many people, including kind of Christian Kaiser, who's here in the audience, has participated quite strongly to this. And I think what's certainly emerging is that the ribosome has a key role in modulating and regulating the fate of the nascent polypeptide. As a kind of try and depict several examples here, but even up to 30% or more of assemblies begin on the ribosome, as is emerging from many of the profiling experiments by people like Bernd Buchau and others. So, a polypeptide chain either interacts with another polypeptide on a neighboring ribosome, or can interact in a co-post manner with an exogenous nascent chain to begin to form its complex. And that kind of makes sense, and has always been something that has always perturbed many people, in that how could a largely disordered chain essentially be released incapable of folding because it needs its assembly partner to fully fold, in danger of many aspects of degradation unless it's strongly protected. But the ribosome provides a mechanism where some of this can kind of happen. And the importance of co-translational folding has been revealed by many people. And kind of I show our example here where, in many refolding, it differs from the refolding of proteins. And that was the second point I was raising there. There's significant differences between co-translational folding and in vitro refolding of a polypeptide. And this talk will describe some of these features. But if we take a simple polypeptide, RAS here, the refolded polypeptide has weak activity, while the nascent polypeptide, this ribosome nascent chain complex, has strong activity and comparable to that of the control. And kind of is the refolded polypeptide, under a vast range of conditions that we've tried, is much more prone to degradation than the respective control, giving us some strong hints that, that there are differences. Actually, this goes further in terms of the differences, if you like, but even misfolding can begin on the ribosome via these co-translation intermediates that we'll hear a lot more of in this slide. And this is actually a slide from my colleague, Lisa Cabrita, at UCL. We've done this work together, where she's built a human cell model of human alpha-1 antitrypsin, and actually managed to see, in many cases, essentially misfolded polymers forming on the end of ribosomes. So you can see these chains forming on the ends of ribosomes. And this kind of makes sense. Up to 30% of ribosomes, up to 30% of nascent chains are subject to degradation processes. The ribosome provides the branch point for folding processes and invariably, therefore, for misfolding processes. This is a very nice study that she's recently submitted to for publication. It's available on bioRxiv. So we wanted to understand this process structurally, and essentially this slide summarizes how we've been trying to think about this. We've prepared stored nascent polypeptides. We use stalling sequences, so there's a natural sequences that, when reached, result in efficient stalling. And kind of our kind of way of thinking about this was to try to use cryo-electron microscopy to look at the main body of the ribosome, but actually, in places where the polypeptide density diminishes, to attempt to use NMR spectroscopy to see whether NMR spectroscopy could give us some information on this highly dynamic, elongating chain. And this kind of is our first structure of this, where we see a two-domain protein, this immunoglobulin-like main filament, folded on the end of its ribosome and attached to the ribosome, tethered to the ribosome peptide transferase center by its subsequent domain, domain 6, that's beginning to sample conformational space. And this is information derived by a combination of cryo-EM and NMR kind of mixed with molecular dynamics to obtain these structures. So a little bit more about the technical elements of this. I was just talking to Ben Schuler about the actual technical complexity of being able to actually do this. The nascent polypeptide is less than 1% of the complex. It's tethered to a 2.5 megadalton ribosome. NMR is the most insensitive of spectroscopies. So we need huge amounts of sample, kind of, of the order of tens of milligrams to be able to do this. We're in a live complex that naturally has a limited lifetime, and kind of this is a post-hoc killer that averaged out at around 100 samples per manuscript. These samples are derived in vivo, mainly from E. coli cells. So we actually purify the samples from E. coli, purify the nascent polypeptides from E. coli. So with that kind of slight lament, I'll show you some of the very, very promising results that emerged from some of these early studies. We can see these fingerprint spectra, and for those unfamiliar, these are proton-nitrogen fingerprints, and here proton-carbon fingerprints with selective labeling. And you can see these are rather attractive spectra, and these are of nascent polypeptides attached to their 2.5 megadalton ribosome. So this was a very promising result, immediately telling us the nascent chain is present, certainly, because we can verify this through a range of biochemical and NMR diffusion experiments, and also the ability to observe resonances tells us that the nascent polypeptide is indeed a dynamic entity. So this means that we can do experiments like this. This is my first experiment actually on residual dipolar couplings. Was actually taught to me by Trevor Rutherford, who was here in the audience earlier. Well, we went on to use this on ribosomes. We can kind of align the ribosomes in filamentous phage, and through these kind of dipolar couplings, we could begin to use these as structural restraints within structure determination processes. Kind of just a quick kind of heads up to the EM. You get these exquisite density for many aspects of the ribosome. We can see here, and even more recently, actually, we can begin to even see the nascent polypeptide, slight blobology perhaps, and kind of wishful thinking, but we can begin to see blobs for the nascent polypeptide kind of folding within the exit vestibule. But actually, the most of folding begins outside of the ribosome for the vast majority of polypeptides. So NMR seems to be a powerful way to begin to look at this, and indeed it kind of has been for many years. And just to give you a kind of idea of this, this is with isolated proteins now, where we take my favorite immunoglobulin domain, 106 residues, and if we truncate this by 21 or so amino acids, and I'll show you the NMR spectrum resultant from this, we'll see a fingerprint here, proton nitrogen fingerprint. And for those who know, this is a disordered polypeptide, has all the characteristics of a disordered polypeptide, but nevertheless readily assignable, means you can work with it quite extensively. But more importantly, if we kind of add amino acids and go towards the full-length protein, we can watch the process of folding. We go from the disordered state all the way to the native full-length protein and several of the states as it begins to transition between these, including characterizing two intermediates at various truncations that we can characterize and readily, straightforwardly determine structures of. So the focus of this talk will be this immunoglobulin-like domain. We've explored several other systems, and this has been the gift that keeps on giving. Essentially, these are the fingerprint spectra of the folded state and the unfolded state of this polypeptide. But actually, what has emerged over recent years is the power of 19F NMR spectroscopy. Fluorine labeling is readily incorporated into this. We incorporate this into the protein by simple amber labeling methods. Can choose our position. We've done extensive studies on choosing the position of this. I'm going to show you a study with a phenylalanine labeled at 655, which has been replaced with a trifluorophenylalanine with the 3 fluorine groups. Now, fluorine has several advantages. It's not present in nature. It has a wide chemical shift dispersion, and you can do some relatively straightforward spectroscopy, which is actually will turn out to be, we've seen to be rather important for looking at the larger complexes here. So I'll show you the spectra of the isolated protein. We've got the isolated folded protein with a chemical shift at this position. We've got the respective protein that's been rendered disordered at this position here, both isolated. So we have a chemical shift difference between these two. And this gives us a playground to play with. And I'll show you kind of the denaturation of this, or part denaturation of this. And this is essentially a 2-state folder in isolation for the full-length protein. We see the unfolded state, we see the native state, but we don't see any other states. Now, I'll kind of wind forward to looking at this on the ribosome now, where we look at different states of elongation. And at the early stages of elongation, we see an unfolded signature here for the 655 chemical shift. We see the folded state signature once it has completely emerged. But at intermediate states of synthesis, apart from these two states, we begin to see key slowly exchanging additional peaks, exchange very slowly with these other states that are certainly stable intermediate states of this protein. So we're immediately seeing that there are, apart from this being 4-state, seems to differ quite significantly from that in the isolated protein. And by kind of producing a set of nascent polypeptides and progressively elongating, so essentially increasing the amount of domain 6 that is synthesized, we can begin to see the process of folding from the unfolded state to the folded state, and we can see these intermediate states forming, and we can actually work out their populations. You see the reduction of the unfolded state, the increase in the folded state, but actually these intermediates, two distinct intermediates, have their rise and fall over the process of elongation. And these are, our initial experiments suggest to us these were partially structured. When we remove the capacity to fold of this domain, we kind of resolve to one peak at the unfolded state, suggesting that this I1 and I2 states that you'll hear more about are at least partially structured. I'll talk a little bit more about this, but we've recently actually managed to determine, this is an extensive set of experiments. We had to exclusively use 19F NMR. So this is where the hundreds of samples that I described earlier come in. We actually managed to look at structures of these folding intermediates of the ribosome. So we have the I1 and the I2 state, and the I1 and I2 have close to 40 or so residues at the end terminus that are detached essentially from the main chain. So you have the core of the protein, this FCED core, but you have this part is actually away from the ribosome, and this other intermediate state has the C-terminal region quite extensively detached. Now, these are well emerged from the ribosome. So these should, in principle, have the capacity to fold. So these are distinct intermediates on a pathway. And I'll kind of show you some of the elements of this. You can see, in particular, kind of compared to the native state, you can see this kind of extensive block of residues in intermediate I1 that are essentially refusing to form part of the core or form part of this intermediate state on the end of the ribosome. And I'll talk more about this a bit later on as to the basis of this. So the question we asked, once we saw some of these data, i.e. 2-state for the isolated protein, 4-state at least for the ribosome nascent chain, is why do these folding thermodynamics and mechanisms differ on the ribosome? This is a key question in the field. People have looked at it in various ways. So I want to kind of very briefly introduce a study that has been published where we try to understand these effects. And so initially we kind of wondered whether interactions of the nascent polypeptide with the surface of the ribosome were actually inhibiting folding and causing formation of some of these intermediates. And again, we kind of developed a very nice NMR, and Ace Castignon in the lab developed a very nice strategy where you could use NMR and use fast exchange principles to begin to understand a nascent polypeptide that is essentially interacting with the ribosome, and as the chain elongates towards its completely released state in gray, we can kind of see this change in signature that was present, revealing that we could obtain a molecular ruler of the types of interactions that were occurring of the nascent polypeptide with the ribosome that inhibited the folding process. However, given that this is the main subject of my talk, we found that this was only able to account for at most 1 kcal per mole of the inhibition of folding. So this interaction with the surface of the ribosome is not the key determinant of the differences on and off the ribosome. Certainly there was some enthalpic stabilization I'll talk more about, but actually it's not the main determinant. So then we went on, simultaneously we went on to trying to use some of our early structures and actually to try to look at whether or not the interactions of the nascent polypeptides, here I kind of show you 2 structures, alpha-synuclein and FLN5, with the proteins within the exit vestibule of the ribosome and beyond, were actually whether they were responsible for the inhibition of folding. So we made a set of E. coli, a set of CRISPR mutants. We're using principles of bioinformatics. We essentially truncated various loops that were tunnel protruding, L24 and L23 predominantly, and we also made insertions within the exit tunnels to try to sort of see what the navigation pathway and effect on the energetics on the nascent chain were. And kind of very briefly, these results again, if I compare this to, we used our favorite friend, 19F NMR, and if we kind of take the wild type at this particular elongation length, where the nascent polypeptide is attached, the domain is attached to the ribosome by 34 amino acids, so at this point it's about 60 or so % folded, we could actually make some significant dents on the extent of folding that we observed, in particular with deletion mutants, which were kind of somewhat to be expected, given that we've made a little more room for the nascent polypeptide, which is rather constrained within this tunnel for it to fold. Some other interesting observations occurred when we started to actually kind of create insertions of, for example, L23, which have led us to kind of some interesting kind of hypotheses of pathways of the nascent polypeptide within the tunnel that we've recently kind of submitted and we're working on. But actually, even despite these observations, the energetic consequences of the interactions and the pathway changes within the tunnel are actually relatively small in terms of their contribution to folding. So onwards, we kind of decided to think about the process and look at the unfolded state somewhat. and to determine, to try to understand more about the unfolded state on the end of the ribosome. So we determined some structures of these, and this is a sort of cut to the chase. Essentially, this is the isolated unfolded protein that we've determined structures of. You can see it's an ensemble of structures, rather compact ensemble. We can see the same structures here on the end of the ribosome. These are experimentally determined structures. And we can actually see that there's significantly reduced propensity for residual structure on the end of the ribosome. And I'll describe that more in a moment. So you can see differences between these states and in isolation and those on the end of the ribosome. How we did this? Well, essentially we used paramagnetic relaxation enhancement. These are highly dynamic states. They're not open to electron density, essentially, or to nuclear Overhauser effect. So we use paramagnetic relaxation enhancement, NMR, whereby attaching an unpaired electron, essentially at various points onto the sequence of the protein. We could use the broadening that results when an unpaired electron comes close to a nucleus and monitor that across the sequence of the emerged domain. And use this NMR together with cryo-electron microscopy structures within of the polypeptide within the exit tunnel and merge the two using molecular dynamics methods. So kind of, this is again saying the same thing. The various places we've chosen across the sequence to be able to do the labeling. We kind of see some of these enhancements. You can see the diamagnetic spectra and the paramagnetic spectra shows some quite significant broadenings across the sequence compared to the diamagnetic spectra. And we can quantitate these, and this is one example of the actual many positions of labeling that exemplify the situation. So if we take the isolated protein and the nascent in black and the nascent polypeptide in the blue bars, and what we're looking at here is the impact of labeling at position 740 on nearby residues. And there's a distance dependence to this, an r to the minus 6 distance dependence to this. And what you see with the isolated polypeptide is that labeling here actually causes relatively extensive broadening, despite this being a disordered state, while the corresponding nascent polypeptide is relatively unaltered. And we can kind of do this from the perspective of look at the impact of the ribosome to help us to determine structures. These are just two of our positions of labeling, where we used CRISPR-labeled ribosomes to place a nitroxide spin label to allow us to get distances from this position here to the nascent polypeptide, the unfolded nascent polypeptide, even though I depict this here on the native structure. So these are, going back to the slides that I showed you earlier, apologies, these are the structures that resulted from this. You can see that the isolated unfolded protein has a range of contacts. I depict the native contacts in black of the native protein. You can see many of these are native, and there are also many non-native, while there are significantly fewer contacts observable in the nascent polypeptide. So it's a significantly expanded species with an increased radius of gyration on the ribosome. These ensembles were initially looked at theoretically. So this is where we could use a range of actually quite powerful algorithms that are emerging to actually begin to understand some of these, to actually begin to give some initial indications of what we're seeing that could then be used to decide on experiments. And if we look at these ensembles, they predict that both the conformational and solvation entropy are significantly reduced on the ribosome, combined by up to sort of 13 kcal per mole of destabilization. And this is mainly seen in areas, you can see these destabilizations are mainly seen in areas where the local radius of gyration increases. This significantly outcompetes enthalpic gains in stability from ribosome interactions and the increased solvation, and predicts the net increase in the free energy of the unfolded state on the ribosome by up to sort of 9 kcal per mole. So these were useful predictions, but of course we wanted to see the impact on the folding. We wanted to kind of determine and measure these thermodynamic parameters experimentally. And in order to do this, we kind of measured folding equilibria of this domain on and off the ribosome. So this is on the ribosome and off the ribosome with a truncation that we've used here across a range of temperatures. You can use the equilibria to actually help us with both determine the enthalpy and the entropic impacts using the Van Hoff equation. You can immediately see that our hypotheses of destabilization are verified by much shallower curves that you see for the nascent polypeptide compared to the isolated protein. You can kind of see, see significantly reduced impacts on the nascent polypeptide, and some kind of quite significant differences between the nascent chain and the isolated protein in both enthalpy and entropic considerations. So the prediction of the entropic penalty being lowered is actually the case. And while enthalpy is also, folding is also less enthalpically favored, we have reduced, rebalanced compensation of enthalpy and entropy, but you can kind of see the differences in magnitude between these two effects. So try to correlate these effects and try to understand the structural impacts of these. So these are now the paramagnetic relaxation enhancement curves repeated for both the nascent polypeptides that I've shown you previously and the isolated polypeptide and the corresponding thermodynamic curves, the Van Hoff curves, the two cases here. And if we actually extend the linker of this nascent polypeptide, so we bring it out towards the more released form, we can actually see that the increased linker significantly decreases the expansion, although less so than the isolated protein. And you can see that correspondingly, the energetics of this emerged nascent polypeptide are intermediate between those of the short nascent chain and the isolated protein. So this begins to establish a relationship essentially between nascent chain structure and the folding thermodynamics. So in the interest of time, I'll slightly summarize what we found from this actually. What kind of actually happens here is that this destabilization of the U state of the nascent polypeptide compared to that in isolation actually rationalizes very, very clearly the appearance of these folding intermediates. These are up to 5 kcal per more stable versus the U state on versus off the ribosome relative to their corresponding isolated protein. This I state stabilization that occurs can't be rationalized. We can only rationalize it to this extent. We've done extensive chemical shift analyses and comparisons by ribosome interactions, but actually entirely by the entropic destabilization of the U state. And we see these intermediates for a range of proteins that we've been working with, and Christian has seen these for several other proteins using his optical tweezer methodology. And these kind of intermediate states that we're seeing actually represent new structured conformations on the end of ribosome. They're fairly ubiquitous, as we see experimentally, on the end of the ribosome, and these states that are too high in energy in isolation are being used on the ribosome as part of the folding pathway. So in the interest of time, I see that this entropic destabilization leads to these persistent intermediates, and in a story that I'm not going to tell you, it protects nascent polypeptides from mutation-induced unfolding. And I hope overall begins to show you that this NMR methodology, compared with extensive molecular modeling, can be used to develop useful tools for the biosynthesis of increasingly complex systems. And begin to help us to rationalize these types of observations. Essentially, when we try to refold the nascent polypeptide, this is the slide I showed you at the beginning, we begin to refold the polypeptide in isolation, we kind of see a significantly reduced activity, while the population of intermediates that occurs during the ribosome in both these two cases allows us to achieve a stably folded protein. Okay, I'll stop there and thank the people in the labs who've done some quite tremendous work over the year. In fact, almost all people are now former lab members, many of whom have achieved their own positions now over the last couple of years. But they've done a tremendous job at this, and I thank you, I thank them, and I thank you for your attention.
Thank you, John, for this fantastic talk. Are there questions? Yes, I see already 1, 2, 3. Yeah.
Thanks, John. Very short.
Hello, Dina. Nice to see you again.
Yeah. So if you assume you would have a multi-domain protein, it would make sense maybe to fold one domain, then speed up in the linker region and fold the second close to the ribosome and so on.
Right.
Do functional data indicate that the ribosome stalls a bit to fold those individual domains close to the ribosome?
I think that is the onset of co-translational folding when experiments such as Hartle and Friedman's experiments, that's what they were showing, is that one domain could emerge from the ribosome, fold, and then the subsequent domain could emerge, and there'd be kind of some sort of inability, for example, in the case of repeat proteins, to have interactions between domains to enable fairly parsimonious folding, or to enable a part of a protein that is useful for folding to emerge before the subsequent domain emerges. But what the nuance here is that these kind of intermediate states, even within isolated proteins that we're seeing for these proteins, and in particular for many negatively charged proteins, these intermediates are the routes by which even these domains are folding. And kind of, Christian may or may not speak about some of his work with elongation factor G, and where he's looked at the progressive synthesis of a domain. I mean, this gets more interesting, essentially, because what can happen is that domains can emerge, and the formation of these intermediates means that you can even expose surfaces that are subject to the ability for so-called post-translational modifications, but actually occur co-translationally in these cases. And some of these intermediates expose these residues that are otherwise buried to be able to do this. Christian?
Yes, just a technical question. Why the choice of a not of a paralytic cage lantanide, but just of an organic?
Of the nitroxide spin label. Yeah, I mean, we have data trying to actually look at essentially lanthanide shift effects, essentially. I forgot what they're called at the moment. Can we try and look at these? And they work similarly to some extent, and we see some significant lanthanide chemical shifts for 1 or 2 systems that we're trying to analyze.
I was not talking about using lanthanides for chemical shift, but just for relaxation.
We've
like the gadolinium, because usually people say that if you have a cage lanthanide, you reduce the internal mobility of the paralytic probe. So I was just wondering whether there was a technical problem in it.
I think the nitroxide spill label has been the label that's been predominantly used in protein folding or relatively straightforward protein folding studies. But actually, I think you've opened up quite a major question, because actually using other paramagnetics to get different angular effects will be much more powerful and be able to get paramagnetic chemical shifts rather than just relaxation. But that's actually something that we're working on to achieve more restraints, because one of the problems is restraints.
Go ahead with the question. Sorry, it will be the last one because we are over time. Yeah.
By me. It's a really amazing platform that you have built there. And my question is a bit related to the previous one. So you have this non-equilibrium process of like chain elongation. So is there a way to actually get that kinetics with any of these techniques? Because I think that it would really...
I think we'll struggle actually with the NMR methodology. We can certainly get dynamics within this. I mean, dynamics from NMR, and that's where people like Fleming and others have been very, very good at this. So we can potentially see the range of states present within any one state. But these are fundamentally equilibrium snapshots. So pathways, excuse me, which is one of the, excuse me, one of the key issues here is whether our static subshots can be made to represent a pathway.
Maybe even just order of magnitude. I mean, how it relates to sort of the time that it takes to synthesize.
I think that you can tackle many of these things using a range of ensembles and try to understand these states. I mean, the ability to discern what we can do, and I'm describing what we can do because what we actually need to be able to discern kinetic pathways are real-time experiments. I mean, I think from experiments such as your optical tweezer experiments, which is why I want to talk to you to be able to look at these. But essentially, in this case, we clearly have two different intermediates that are not able to be prescribed as a sequential pathway in this case. In other cases, we can do that with Tixin, for instance, because it just is a nonsensical thing that you would have the A and B strands detached in one case and only the E and F strand, EFG strand detached in another case. They would have to be inferred as parallel pathways, but it's an inference and really needs more real-time experiments to sort of solidly be able to say, this is the pathway. That's a good question. And
amazing, never
Thank you, John. And during lunchtime, people who couldn't ask a question now, please approach John. Thanks.
Thank you.
Thanks.